Evolution describes how life changes once life is here. It's not a theory about how life originally arose or why the whole universe exists. Yeah.
I was wondering, does the entire process move at one steady rate, or can it sometimes accelerate and decelerate with different environments and different during evolutionary time?
We're going to come back to that point during the second part here, because we actually can see jitters in the fossil record of evolutionary change. It does happen with different rates at different times, sometimes fast, sometimes slow. Again, there's nothing in the theory of evolution that says it has to be particularly fast or particularly slow. In fact, we know genetic mechanisms where major genes can have big effects or other genes can have small effects. That could create rapid evolution or slower evolution. There's no particular rate that evolution requires, and in fact, a range of rates are seen in the fossil record and in experimental systems. Okay, we better come back to the second section. Happy to take more questions here in a minute. Okay, so let's come back to some of these scientific debates about the way selection could or couldn't work. One of the major debates was whether natural selection would be able to let an interesting new variant affect traits within a population. So as I said, people believed in blending inheritance at the time the origin was first published. The problem that was pointed out was, well, suppose a little tiny drop of black ink appeared and was dropped into a giant vat of white liquid like milk. Okay, a tiny drop of black ink in a giant vat of white milk has no effect on the overall color of the vat. It's still just blended out, and the population would stay predominantly milky white. Okay, sorry, it's even confusing to try to explain, because we now know so much more about the genetic mechanisms that actually exist that blending inheritance clearly doesn't happen. The Origin was written before any detailed knowledge of genetics, so those kinds of ideas were out there. But when Mendel's laws were rediscovered in 1900, it was clear that variants aren't just lost by blending. In fact, variants exist as stable genetic changes that exist on chromosomes. Those stable variants are sometimes hard to see in populations. Variants can be recessive. But once a mutation arises, it can increase in frequency simply by being transmitted to more and more offspring. We made extensive use of Mendel's laws yesterday in talking about the genetic architecture of corn and teosinte evolution, stickleback evolution, and the evolution of color forms in rock pocket mice. Sean actually showed you a simulation based on Mendel's laws and population genetics of how a rare variant, when it appears, doesn't just get blended in. In fact, a small selective advantage, even a 5% increase in the number of offspring that survive and reproduce from a black variant, will lead to a rapid increase in the frequency of that variant over time. So at the population level, the variant doesn't blend. It appears, and if it generates more offspring, it will actually increase in frequency as the white form decreases. Now, this data was based on a simulation, but I'd like to compare the results of that simulation with the results of the actual frequency of pelvic forms that are seen in the fossil record that we just described for pelvic reduction in sticklebacks. So here, the line graphs represent the reduced form of pelvis, originally as a rare variant, that sweeps to fixation in the successive layers that can be dug up out of this Miocene lake bed. I hope you can see that although this is simulated data, and this is actual data, that the form of the curves are strikingly similar. So variants don't blend when they appear. In fact, they can spread through populations using laws of both Mendel and population genetics that are now well understood and that are also confirmed by observations in real systems. Okay, finally, how about this last problem? Do animals simply look too different to come from common ancestors? This is, again, an area that Darwin tried to address in The Origin. He's got this passage. All living things have much in common. similar germinal vesicles. We see this also in that the same poison can similarly affect both plants and animals, or that the poison secreted by the gallfly will produce growths both on wild roses or on oak trees. Darwin was trying here, but you can tell that at this stage, the knowledge of biochemistry and molecular genetics was so rudimentary that the arguments for the interrelationship of all living forms was still very circumstantial. Since the origin of species, pathways for the synthesis of DNA and RNA and proteins and lipids and carbohydrates have now been worked out in detail. Turns out most bacteria, plants, and animals share fundamental metabolic pathways. They all use ATP for energy. They have lots of other biological characteristics in common. You can actually take enzymes out of one organism like a mammal, mix them together with bacterial extracts, and the enzymes and bits and pieces from different organisms are compatible and can catalyze these reactions. Most importantly, and one of the triumphs of 20th century biology, the molecular basis of heredity has been worked out in detail. So the structure of DNA was discovered in 1953. That led to the identification of a universal code for converting DNA sequence into amino acid sequence in most animals. Same sorts of DNA, same sorts of genetic code, whether you're a virus or a bacteria or a plant or an insect or a mouse or a human. Genes from one organism can actually replicate and function in another, a striking confirmation of the common biochemical and genetic heritage of all living things. Finally, very different looking animals are actually built using similar genes and pathways. One of the most striking examples of this was originally found in research in Drosophila, the fruit fly. Drosophila has a whole set of key developmental control genes called Hox genes. These genes occur in clusters along the chromosome, and the expression pattern of the genes in the embryos is related to their position along the cluster. So the blue genes here at the one end of the cluster are expressed at the head end of the embryo that's shown there in the bottom right. The colored genes in the middle are expressed in the middle of the embryo, and the green genes at the end of the cluster are expressed in the tail region of the embryo. So those genes aren't just expressed at different anterior, posterior, head-to-tail positions. They're also required for the formation of the particular tissues that you find from the head to the tail of the adult Drosophila. So if you make mutations in those genes, you change the development of the characteristic tissues that should form along the anterior-posterior body axis. Well, surprisingly, although animals look very different, mice look very different from fruit flies, their anterior-posterior body axis is built from the very same sets of genes. So mice and humans, other vertebrates, have sequences that are closely related to these Hox genes of flies. The Hox genes in mammals also occur in clusters. The clusters are also expressed in patterns that are related to anterior-posterior development. Blue genes at one end of the cluster, they've actually duplicated, so there's more copies in mammals, but blue genes at the end of the clusters are expressed in the head regions of the embryo, shown at the lower left. The middle genes are expressed in the middle part of the embryo. The green genes at the end of the cluster are expressed in the tail region of the embryo. And just as in fruit flies, if you make mutations in these key Hox genes, those mutations alter the formation of the characteristic tissues that would normally form at each position along the anterior-posterior body axis of the mouse. So it looks like the head-to-tail axis of animals, even that look completely different, are in fact built by an ancient toolkit of these key developmental regulators called Hox genes. Similar toolkit genes have been discovered that control a range of other structures in diverse organisms. So the Hox genes for the anterior-posterior axis, other sets of genes for the dorsal-ventral axis, other toolkit genes for the left-right axis, and other toolkit genes for the formation of particular body tissues. Let's give an example of one particular tissue, eye development. So eyes, very interesting structures found in lots of different animals. They look quite different in different animals. So that's a human eye and a Drosophila eye. Human eyes has single light-gathering organ and lens. In contrast, insects have these compound eyes, thousands of little light-gathering organs all in a little